Oops. I'm going to go ahead and get started. Normally, I will try to, go over the slides with the PowerPoint stuff. Going over like the most missed topics and things like that. For these chapters, for chapters 15 and 16, I have quite a lot. Let me see. Let me turn this off. Make sure this is off. That is okay. I'll deal with this later. Okay, so I'm going to do a whiteboard thing this time. I'm not going to do the slides from the PowerPoint. I just have way too much going on a personal level with things currently and just in my life right now. I had the funeral yesterday, but there's a lot going on in family stuff right now. So I'm going to take the time to go over the entire immune system and the processes of the immune system. I'm going to leave the PowerPoints with the most missed topics. Hopefully I will have covered that stuff, right? Hopefully I will have covered all that most missed topic stuff, and we will have gone over that in this explanation. But if there's anything for anybody going over the PowerPoint slides that would have been used in this talk, and you can't figure it out for yourselves, then please let me know before you get to that point. But for today, what I'm going to do is outline the basics of the immune system, and then I'm going to draw the process out as well. So we'll walk through all of that stuff with talk as well as writing it out on the whiteboard. And then I will, after drawing all this out on the whiteboard, share the whiteboard as well. So I'll have to change the settings with that apparently, but just FYI, that's all going to be what we're going to be doing here today. Got the message from, there we go. Okay. All right, so the immune system. So first things first, gosh, come on. Let me change this so it doesn't keep switching orientations on me. There we go. So first things first with the immune system, I'm going to go over the cells that are involved in the immune system. So we have the pencil. Okay. We have the, in the immune system, the leukocytes. Leukocytes is just another word for white blood cells. All right, within the category of our leukocytes, we have two different versions of our leukocytes, granulocytes and agranulocytes. Before I get too far into this, Heather, you're the only person that's on here. Can you actually hear me when I'm talking? Because it's not popping up with my icon when I'm talking. Are you able to hear me? Because that's the only thing I'm worried about, is that it's not actually recording what I'm saying. Okay, awesome. Okay, great. I just wanted to verify that because that would suck if I was doing this and it's just me writing things. Somehow my pencil isn't charged either. That's great. Hopefully that doesn't die. Okay, so our granulocytes, basically we will break down as the fills. First, actually, let me reorganize this just a little bit because I'm going to put a random outlier first, which is our mast cells. And then our other cells, which are going to be our fills. So this will be our, break these further down. This is going to be our eosinophils, our basophils, and our neutrophils. All right. For our agranulocytes, we kind of have two major categories with that as well. So our agranulocytes kind of break down into two major categories, which is going to be our monocytes which are in the blood. And that when these go into the tissue, can become macrophages and dendritic cells. For the agranulocytes, we also have another group called the lymphocytes. And that our lymphocytes are kind of into two categories, the T cells and the B cells. And the T cells, there's two different types of T cells. There's the cytotoxic, and that the cytotoxic are CD8 positive. And then the T helper cells, which are CD4 positive. The T helper cells will then break down even further once they're activated into two forms. Let me spread this out just a little bit better so that we have some division going on here. The T helper cells can be TH1 or TH2. So I want to be very clear about this, which is to say that TH1 cells, while they govern and activate these guys here. So these guys are actually going to be involved in activating those guys. That TH1 cells are CD4 positive. CD4 positive. Because they are TH cells, even though they govern the cytotoxic T cells, they actually activate them and help control their response. They're the captains. So these guys are our captains. That these T helper cells, get a color here. T helper cells are CD4 positive. Our CD8 positive guys, on the other hand, are only our cytotoxic T cells. So the guys that are actually actively cytotoxic T cells have CD8 positive markers. Our helper cells have CD4 positive markers. And so either your TH1 or your TH2, the TH1 is the captain that governs the cytotoxic guys getting activated, but they're still CD4 positive. And this is important because we're going to talk about how that gets activated in a little bit. But yeah, so, okay, so scrolling down, oh geez, undo that. All right, so are those one type of lymphocyte, the T cells, and then the other type of lymphocyte is the B cells. And now when the B cells get activated, plasma cells which are the effector versions of the B cells, they make antibody. All right, and when they make antibody, the first type, as I'm scrolling down a little bit further with this, the first type of the antibody that they make is IgM. Then we will class switch to either IgE, which we usually see in parasitic responses, IgA, which we see in mucosa, or IgG, which is our workhorse antibody. We make the most of IgG, and it works the hardest for us. So those are the main antibodies that we can make. So I'm leaving out one type of immune globulin. That is on the B cells, their receptor is IgD. That's the B cell receptor. So this IgD is not secreted into the bloodstream and everything like that. Just the other guys are. Okay. So the Th2 guys are the ones that are going to be activating the B cells. Okay, so we come back up to our T cells and we look at CD8 and CD4. CD8 sees MHC1 that's bound with antigen. CD4 sees MHC2 that is bound to antigen. All right, and so MHC1 presents inside antigen, and it is found on all nucleated cells. MHC2 presents outside antigen. And the only cell, this is really getting out of hand with my handwriting here. The only cells that can present the outside antigen, the only cells that have MHC2 are antigen presenting cells. Are antigen presenting cells. I'm going to get a highlighter and highlight those guys. It's a pencil. I want a highlighter. Let's do these guys in yellow. Our antigen presenting cells are our B cells, our macrophages, and our dendritic cells. Technically the monocytes too, but these are our biggies here. So our, because the other guys are derived from the monocytes, right? So they're the same, kind of the same thing. So these guys are all, we'll put this in a different color. We'll put this also in, not yellow because that's too, we'll do this color. All right, so those are our antigen presenting cells. Two of these antigen presenting cells are also phagocytes. So let's mark our phagocytes. We'll do them in green. Not how I meant to do that. I'm gonna do that as a highlighter. We'll highlight them in green. So macrophages and dendritic cells and monocytes, as well as neutrophils. Green, in this case, will be our phagocytes. So we can see here that our phagocytes are going to be our neutrophils, monocytes, macrophages, and dendritic cells. Our antigen presenting cells will be our monocytes, our macrophages, our dendritic cells, and our B cells. So our deviants here will be our neutrophils on the phagocyte side. They are phagocytes, but they are not antigen presenting cells. Whereas B cells are antigen presenting cells, but they are not phagocytes. And the way that the B cells get the antigen is because it bound to their receptor on IgD. And then it brings it in from that receptor. It's not phagocytosis, it is receptor driven interaction. And so that's a different kind of process going on there. So it is not a phagocyte. The B cells are not phagocytes, but they are still antigen presenting, and they present the antigen that bound to their receptor, the IgD molecule. The monocytes, macrophages, and dendritic cells are phagocytes. So they are going around and just eating up things in their environment and then presenting that outside antigen on their MHC2 molecules. And that's how they work as antigen presenting cells. Neutrophils are phagocytes, but they're just going around and eating things up and digesting it and then leaving all that waste behind. And so that's how they're going to be phagocytes, but not antigen presenting cells. And so this is the breakdown of those cells and how they're going to be working in the immune system kind of together in their roles. And so this is the gist of that. We're leaving out an aspect of the immune system that doesn't have to do with the cells, and that's on the innate immune system side. So let's switch over to my black pen again. On the innate side, we also don't want to forget about inflammation, and that, that inflammation is going to be basically triggered by our granulocyte response. And so all of these guys over here, so I'm going over to the left a little bit, all of these guys here, I guess you guys are seeing my actual screen and not the whiteboard, so you're probably keeping up with me just fine, but let me change this just a little bit, sorry. Okay, cool. All right, these guys here, these cells that are involved is our granulocytes, but these guys are signaling inflammation. With their response. So those guys signal inflammation. And what else can signal inflammation? Complement. And that complement, that set of 50 proteins in your bloodstream, can lead to the signaling of that inflammation. Actually, let me draw the arrow a different way. Let's do it like this. Complement could signal inflammation, but complement can also lead to we're going to do the membrane attack complex. Let's go ahead and do that. The membrane also known as the MAC, where complement protein, they all come together and polymerize. Once you activate complement, you initiate the cascade. They then can polymerize and then generate the construction of the membrane attack complex. And that this hole that might be in a whole bacterial cell can cause leaking out of the contents of the bacterial cell. That's the membrane attack complex. Complement can also signal inflammation, but complement can also have a role in helping with opsonization. Opsonization is molecular handles that help with phagocytosis. Now, this is part of our innate immunity, that complement pathway. How do we activate complement? Well, I'm glad you asked. So coming over here to the left, with our complement pathways, we can have 3 pathways: the classical, the lectin, and the alternative. And that all of these will activate the complement pathway. The classical pathway I can hold on to my iPad here. It's sliding all over the place. All right, the classical pathway is activated by antibody. The lectin pathway is when a protein called lectin binds sugars that are on the surfaces of bacteria called mannose. We'll just put bacterial sugars. Touching my screen weird. Bacterial sugar. That activates the lectin pathway. And then the alternative pathway is activated by repeating patterns, kind of like PAMPs. So this will be things like LPS or endotoxin. That these things will trigger the activation of the complement pathway, and the complement pathway can signal inflammation. It can lead to the membrane attack complex, or it can lead to opsonization. Interestingly enough, our antibodies over here, one of the things that they can do is lead to opsonization, right? We'll talk about the other functions of our antibodies as well, just since we've already started on this, that can lead to opsonization. It can lead to coating. It can lead to toxin neutralization. So we call that antitoxin. It can lead to general neutralization of docking, basically. We just call it neutralization. Opsonization, coating, antitoxin, neutralization. We also have agglutination. Put these down here more. Draw the little lines to them. And I'll put some descriptors with these too. We'll kind of outline them a little bit, just that we're aware of everything that's going on with this stuff. Opsonization. And in addition to opsonization, one other thing that antibody does, of course, which we already have over here, is the classical pathway of complement. It's all the same thing. So those are our 6 possibilities of the actions of our antibodies. Activating the complement pathway, which complement itself can signal inflammation, can do the membrane attack complex, and complement itself can aid in opsonization. Antibody can also add in opsonization. So it can either directly lead to opsonization, helping out with grabbing onto things by the phagocytes, or it can activate complement, and complement can help with opsonization. So we've got 2 different targets for helping out with molecular handles for our phagocytes to grab onto, either triggered by the antibody directly or the antibody triggering the complement pathway and the complement leading to the handles. And so in addition to by activating the complement, you can signal inflammation. So antibody can signal inflammation, but it can also initiate that membrane attack complex, which is like the big goal of complement, right? It's the big role that complement really plays with that. With antibody, when we have coating, the coating is just covering and blocking in general actions. And interactions with the environment. The antitoxin binds to the toxin, so it can't do its job. So these toxins are what are secreted by the actual bad guy, and that's what causes damage to the host. So we're blocking the damage being done by those secreted toxins. With neutralization, we are very specifically blocking the docking molecules. Switched over. On viruses, this would be our spikes. And then agglutination, scrolling back down here a little bit to the right, that the agglutination would be causing the bad guys to stick together and then inhibiting their function by doing that. H, I promise. All right, so that's our general function of our immune system. If we scroll out and zoom out on that, we can kind of see how these things have gotten outlined and then interconnected with one another and seeing that. Also on the inflammatory process, that all of these guys that can signal inflammation, when we are talking about signaling inflammation, I'm just going to break that down just a little bit more, that this is going to lead to our effects of inflammation of our tumor, rubor, calor, and dolor. We know dolor is pain. We've said it a million times in class at this point. So rubor and calor kind of both come from the same thing, increased blood flow to the area. I want to change this just a little bit. Hold on. Give me a little more space here. Tumor, calor, and dolor. So dolor is the pain. It's from signaling just the nerve endings in the area. Calor is the warmth. Rubor is the redness. And tumor is swelling. The redness and the warmth are basically caused by the actual increased blood flow to the injury area. Come on, pencil. Oh, you didn't die, did you? No, okay, good. Increase blood. It's leading to the redness as well as the warmth. The swelling is caused by exudate. Why does this look like that when I type that? Write that. Some of these, sometime I'm going to write this correctly. Exudate, which is fluid from the blood into the tissues. And on along with the fluid, of course, comes all of your white blood cells and the clotting factors and healing molecules and everything like that. Okay, so that's the exudate, and that the buildup of exudate in the tissues leads to edema. When we talk about calor being a sign of the inflammation, that calor is that warmth. One aspect of that warmth is the increase in the body temperature, which is fever. Let me write it off to the other side because this is going to take up too much space for me to categorize out all my fever stuff. Okay, fever. What did that just do? Okay, here we go. And that fever itself we can sort of branch off from that. We have effects of fever that are going to increase self function, increased host function, we'll say host function, or decrease pathogen function. And we started going into this path of like creating the brain bubbles or whatever, but that's okay. That's okay. So increasing our host function and the things that happen along with that, there's two things that we kind of talked about there, which is kind of increasing our metabolism of our cells. As well as increasing the growth factors. What is happening? And targeting of lymphocytes. And we'll just say white blood cells in general, because that is true. All the white blood cells. Okay, for decreasing our actual pathogen functions, we are going to basically eliminate availability of iron. So we're gonna normally we would let it float around in our actual bloodstream, but instead it's going to be kept away into the actual, away from the actual bloodstream so that the microbes can't get to it, and they need iron too, just like we do. And then the other thing, other than eliminating the iron availability, the high temperature literally inhibits microbial growth. Okay, so now when we zoom out, we really spread out our inflammatory responses. That's okay. We can start to see the big picture of our immune system and the different cells and all of the functions that are going to be going on with that. So I want to be very clear that complement did not come from a cell, right? It just came from proteins that are in the bloodstream, and that's why they're not on the cell side of the list of things. Okay, so we've got this as one outline, one way to look at all of those components of our immune system and how they're going to function all together in fighting off our pathogens. So now I'm going to draw out the actual cellular response from these guys. So let's get into it. Hopefully this won't take too long because my pen is dying and I didn't charge it last night. That was my fault. So we have antigen in an area. This could be a whole microbe. It could just be part of the microbe, and it doesn't matter. So this is our actual antigen. And then we will also have neutrophils come into the area. And they're granulocytes. So hey, let's give them some granules inside there. So that whenever these guys come up, they look like they have like a sort of like segmented sort of nucleus that's connected up to one another. This is just what they would look like if you saw them in the actual microscope. And so that what these guys do is they'll come up and actually eat up the antigen, and then it'll get digested by phagocytosis. And then they will poop out the parts. The neutrophils got called into the area via chemotaxis. They got signals from damaged or infected cells in the area, basically calling for help. And they literally followed the chemical signals like cookie crumbs like in Hansel and Gretel, following those cookie crumbs to this site. And that's how they got brought here. They were in the bloodstream and they followed the signals and then moved by diapedesis out of the bloodstream and into the actual tissue sites. We'll write a little bit of that summary out here. Okay, so now our phagocytes, our neutrophil specifically, which was blue, has moved in here and it's eaten all this stuff up. Next, we can bring in our macrophage. Also called in here from chemotaxis, and moved in here via diapedesis. Remember that when it was in the bloodstream, it was a monocyte, but now that it is in the tissue, it is a macrophage. And so now these guys can come in and eat up this stuff, these littler particles, by phagocytosis again. And that what they have eaten up, they are going to present on, come on thing. A molecule that we will call, starting to realize I'm going to run out of colors pretty quickly. MHC2. So remember, MHC2 is major histocompatibility complex 2, and it will present outside antigen. This will be recognized by a T cell receptor. Sure, purple, I don't know, we're starting to run out of colors already. We'll put TR for this guy. And so this is a T cell. And if we remember by looking up here, T cells that recognize MHC2 have CD4. If they have CD4, they are T helper cells. So here we've got MHC2. We've got our T cell receptor actually recognizing the antigen itself. So it's a very specific interaction. And I want to be clear about this too, that this MHC2 can present any outside antigen. It's very non-specific, but this T cell receptor only recognizes this specific antigen. So now our T cell receptor over here is seeing that, and it has a molecule on here that we mentioned just a second ago, which is CD4, and that CD4 only sees MHC2 presenting outside antigen. MHC2 presents antigen from outside. MHC1 presents antigen from inside. And the T cell is the only type of cell that can see MHC. So T helper cells have CD4 that sees MHC2. Cytotoxic T cells have CD8 that sees MHC1. B cells do not see MHC. Okay, so this is happening. And then when this whole situation happens, let's switch to a highlighter really quick. Let's do, I don't care what color highlighter. Purple, sure. That once this happens, this will lead to activation. Then that T helper cell, when this activation happens, the T helper cell will then proliferate into a whole bunch of T helper cells, just very nondescript T helper cells. Oh my God. T helper cell. Okay. And that some of these will turn into memory. Some of these will turn into regulator. And then these will be our effector cells. Some will turn into TH1, and some of them will turn into TH. I forget that I can scroll over more. TH2. Okay, so now we've got our actual captains here. And these guys are going to regulate the response. The TH1 regulates the inside. And the TH2 regulates the outside. Okay, so how's that going to work? Well, when you have an infected self cell, so we'll have our two different responses here. We'll have the inside and how we're going to actually attack the pathogen. This is our question here. How do we actually attack the specific pathogen? And then we're going to have an inside response and an outside response. Okay. Starting with the inside response, we'll have an infected self cell. It's got, virus inside of it. It's going to put that virus on the outside of itself on an MHC1. Then we have a T cell receptor. That recognizes that specific antigen. And this time, because we've got a T cell that is recognizing MHC1, this has to be a cytotoxic T cell. And that marker that's going to recognize this MHC, which is MHC1, is now going to be CD8. This is our CD8 positive cytotoxic T cell. And that this guy cannot activate without captain's go ahead. And so this captain up here will allow us to activate that response. And now this cell is able to once we've activated that guy, we can make our cytotoxic T cell response, which is by making perforins and granzymes The granzymes digest the cell contents and the cell itself. And the perforins poke holes. And that these guys are going to work together to kill the targeted infected cell. All right. And so that's our inside response. Let me zoom out a little bit. Now let's do the outside. For the outside response, let's zoom in again just a little bit. What we will have is we will have a B cell, and it'll have a receptor on it, which is called IgD. And then that's going to find antigen that it encountered outside in the lymph nodes. So we're in the actual lymph node and we're filtering out that lymph. But this guy is then going to take all of this thing and bring it into the cell and then post it out because it got it from outside on MHC2. So these guys present on MHC2. They do not react to another cell presenting on an MHC. They are an antigen presenting cell. They're actually taking antigen and presenting it on the MHC2. And so that's going to present that here. And yet again, once again, the same old good old friend, the T cell receptor recognizes that specific antigen. In this case, this is a TH2 that has already been activated. If this guy had not already been activated, he cannot yet send signals to activate the B cell. But this guy's already been activated because it previously saw a different antigen presenting cell presenting antigen on MHC2 from outside. It got the signal, it proliferated, it differentiated, and its effector Th2 cell can now activate the B cell. And now our B cell, because it has been activated, the B cell is going to turn into three different B cells. Well, it's going to turn into a whole bunch of them. And those are going to turn into three different categories. So these would be three different B cells. Whatever, there's a whole bunch of them. We'll make millions of copies. And then these guys will differentiate into memory B cells, regulator B cells, and then effector B cells. Which are also known as plasma cells. All right. The memory cells just circulate around and will wait in the future to re-see the original antigen, and they can react immediately and immediately turn into effector cells and immediately start cranking out antibody. However, our regulator cells are there to make sure we're not overreacting to things, and the actual effector plasma cells, these guys are going to make antibody. Which we talked about at the top already, so I'm not going to go over the different classes of antibody again, but we talked about the different classes of the antibody and the roles of antibody in general up in our original outline. And so this is how all these processes will work together in order to make all this happen. Now, if we come back to our B cell interacting with our Th2 cell, and our Th2 cell is able to activate it, if our B cell is acting as an original antigen presenting cell, coming into contact with a T helper cell that has not been activated and proliferated and differentiated into Th2, then that B cell has to wait for that naive T helper cell to go through this process that we went through up here, all of this process, in order to create the captain, the Th2 down here, that this Th2 is the one down here that's able to activate. But if it's not been activated yet, then the B cell has to wait for it to get activated, then proliferate, then differentiate, and then the Th2 proliferated differentiated version that's been activated can then activate that original B cell. Then the B cell can proliferate and differentiate and start making antibody. So now you can understand why the immune response takes a week, because we've not only got to wait for the captains to be made first, then the captains can go out and activate your inside response or your outside response. But the captains have to proliferate and differentiate first in order to basically drive our inside and our outside responses. So our first thing first, activate the captains. I know it is a lot. I tell you guys, I learned this in multiple semesters, and this is being given in basically 2 chapters. All right. Wow, I really zoomed out far on that. All right. And so that is my general explanation. And so being able to take your time to be able to walk through basically everything that I did down here and understand all of the steps on how each one is getting activated and how each one is going to act in each response. And then knowing the general things, like if I ask you on the test, like we had on the quiz question, and a lot of people were getting confused about this, which type of MHC marker does a Th1 cell have on it? You should be able at that point to say Th1 markers, Th1 cells, while Th1 cells are the captains that govern the inside response, the captains always have MHC2 because they had to get activated by outside stuff. The captains are T helper cells. T helper cells have CD4. CD4 recognizes MHC2. MHC2 presents outside antigen. MHC2 is what presents outside antigen and is only found on the surface of our antigen presenting cells. Our antigen presenting cells can be B cells, they can be macrophages or dendritic cells. The macrophages and the dendritic cells are phagocytes that are also going to be able to act as antigen presenting cells. Our other phagocyte that commonly phagocytoses things is our neutrophil, but it's a phagocyte that breaks things down and doesn't antigen present. When it breaks things down, it just leaves things in the environment that the macrophages of the dendritic cells can pick up and present as antigen. If they don't pick it up, it could potentially migrate into the lymph. If it migrates into the lymph, then the B cells that are in the actual lymph nodes, the B cells will be able to pick up the debris or the pathogens that made it into the lymph and interact with it on their IgD receptors, then bring that in and present it on MHC2, the outside receptor. That is how they're going to act as antigen presenting cells. So that's the antigen presenting cycle where we can activate our captains. The captains are always activated by outside antigen, always. The T helper cells are activated by outside antigen, period. Even if you are a Th1 cell, you got activated by outside. The Th1 cells now help mediate the inside reaction by going and interacting with any of those cytotoxic T cells. The cytotoxic T cells are the ones that interact with MHC1. MHC1 presents the inside antigen. The T cell receptor sees that specific antigen. The CD8 verifies that MHC1 is presenting it, but the Th1 cell, the helper captain that got activated from the outside stuff, can now give the activating signal for the inside stuff to happen. So this is my summary of everything that goes on in all of this. I know that right now it's going to take some processing probably, like again, comparing this to what we learned about in lecture to making sense of all of this stuff. And believe me when I tell you, this isn't even the tip of the iceberg, but this is the general stuff of what goes on with the major aspects of our immune response for chapters 15 and 16. So having gone through that, Heather, since you're still here with me, do you have any specific questions for now for this stuff? Okay, so I'm going to leave this and feel free to either watch over this video or try to create your own recreation of everything that I've done here and try to make some sense of it somewhere. And if you get any questions about any of these processes or you want any help with that, you can ask me. You can also, don't forget, you can always ask for ChatGPT to break things down for you as well. That is perfectly acceptable and be able to try to follow it from that, because ChatGPT might word it in a different way that makes more sense to you, that I'm just limited on how I think about these things, unfortunately. So if you don't have any further questions, I'm going to go ahead and just conclude all of this. I'm not going to go over the slides for chapters 15 and 16. I feel like that kind of stuff kind of goes over all of that. The Kahoot, so for a chart that actually organizes medications by what they do, I don't have a chart like that. I think that's a good suggestion. I'll try to put a chart like that together this weekend and get that stuff out to you guys this week. So I'll get a chart like that. That's a great idea. And I actually thought about something like that earlier this week and I just completely slipped my mind. Let me put together a chart for because we kind of have the study chart that's already kind of built a little bit for our chemical and physical control methods. I'll try to make a simplified chart for that as well. And then I will make a chart of the medications. And then this is the chart that you guys are going to get for chapters 15 and 16. It's hard to break it down because they all interact with one another, but we've kind of got our little outline version at the top and then our cell interactions at the bottom to try to break that down. And then we have chapter 17 that I'll make a chart for that. Then it'll make sense why there's a chart for that. And chapter 18 also, well, I'll make a chart for that. So I'll make charts for each of those and make those available for you guys for studying. And I'll definitely get those to you this week. I can't guarantee that it'll be on Monday or Tuesday, but I will try to get it to you at least by the end of the day on Wednesday. And I'll get them to you gradually as I get them drawn up. But I think those are great ideas. These are all very, at least chapter 11, 12, and 17 and 18 are very friendly for charts like that. Chapter 16 and 15 and 16, the innate immunity and acquired immunity, it's not as chart friendly. But yeah, I don't mind making those charts for you guys for 11, 12, and 17 and 18. Those should be pretty straightforward and at least help organize those ideas. That should help you guys with that. And if you have the papers from the tutor from the SSOC, or if you know anybody that has those papers from the tutor from the SSOC to share that stuff, there is some changes that are happening with how they're going to be offering those study sheets, but they should still be very helpful with how they prepare for the exam. So if you have that stuff or you know somebody that has that stuff can help you with kind of orienting yourself with the questions on the test as well. Because I've looked at that study sheet and it has some good stuff on it, is why I bring that up. So, okay. Yes, so that'll be my homework. Charts for chapters 11, 12, 17, and 18. And I will get those to you guaranteed by the end of the day on Wednesday this coming week. Sound good? All right. Thanks for stopping by, Heather. And anybody else that's watching this on the recording, if anybody that watches the recording, just let me know if you want any more clarifying on anything specific, and I'll be happy to take that time for you. And everybody have good rest of your weekend, and I will see you next week.